everyone, my name is Taina. I am a rising senior at the illustrious Tuskegee University. In the spring of 2023, I will be graduating with a Bachelor of Science in Chemistry. This summer, I participated in the UF SURF program in the Ching Medicinal Chemistry Lab. Let's take a peek at what I did. My project this summer is about identifying the presence of an inflammatory marker known as cyclooxygenase 2, or as many refer to it as COX-2, in A549 lung cancer cells. The cells utilized were treated with NNAL, a tobacco-specific carcinogen. The purpose of PCR, or polymerase chain reaction, is to detect the expression of specific genes. Genetic analysis requires a lot of DNA. PCR is a quick and inexpensive method that amplifies and copies the DNA expressed in the samples. First, I would isolate the RNA. Next, create a complementary DNA. And finally, amplify this DNA strand to investigate not only COX-2, but other proteins along the inflammatory pathway, such as MPGES1, an upstream signal of COX-2, and EP2, a downstream signal of COX-2. When it comes to Western blotting, many of my days consisted of cell protein isolation and running a bicyclinic assay. Before we could move on to separating the proteins through gel electrophoresis, transferring the proteins onto a membrane, and staining them with antibodies, the BCA assay ensures the level of protein present within the samples is high enough to potentially detect the desired protein. In our case, this is COX-2 expression. The darker the well, the more protein is present in the sample. One thing I appreciate about this experience is my new understanding of the hours and hours behind the scenes of research that do not go noticed in the final results. This may be in reading literature about fragmentation for mass spectrometry, optimizing thin layer chromatography solvent solutions, or TLC, or undergoing trial and error to find the best method for high performance liquid chromatography, HPLC. Some of my favorite memories at UF beyond the lab consisted of hitting the salsa dance floor con mis amigas, biking through Lake Wahlberg, eating at Harry's in downtown Gainesville, or spending time with the UF Haitian Club Grill. Now that I have undergone part 2 of behind the scenes to graduate school, my confidence in my capabilities to pursue a PhD have grown. I would like to thank my mentors Bree Freeman, and Jessica Mamalapali for being patient with me and transparent about what it takes to pursue a career in research. Thank you to my PI, Chris Ching, and the College of Pharmacy for opening up your lab and pushing me to fully take advantage of this experience. Thank you to UF and NIH for providing the funding to participate in person. Lastly, I would like to thank the Provost, Joe Glover, and everyone else in my life who has supported me mentally, financially, or emotionally. I would not be who I am without you all.